Okay, so now here we're going to look at how meiosis works. So we're going to start with we'll do this one, the DEF chromosome, and then the homologous, or like the second one. And then we have I already know that one. What about FGHI, a blue one, and a green one? And then JKL. Okay, so right now, this is just a regular diploid cell that has two of each kind of chromosome. So all of it, like if this was pretend a human, for example, every single cell in this organism's body would be like this. It would have two of each kind. This is diploid. And you can see how there's two of every allele. So when we do genetics, we talk about homozygous dominant or heterozygous. These are also linked genes. So now, if we're starting meiosis, we ultimately want to cut this DNA in half. We want to make sure that each gamete gets just one of these. And so this is actually Mendel's law of segregation. Mendel's law of segregation says that alleles are going to separate during meiosis or during gamete formation, so each gamete will get one. So that's Mendel's law of segregation. And when we talk about later on independent assortment, how these separate into gametes is independent of how these separate into gametes. These could separate like this, or they could separate like this. These are not linked. These are independent of each other. Now these two genes are linked and would inherit together. Okay, so now, before, this would be like a cell in interphase or in G1. So if this receives a signal to divide, we need to duplicate the DNA and go through S phase. So this is where the sister chromatids are going to be made. So when we go like this here, this is identical. Do you guys want to put your matches? So here we have, we're missing the green. Okay. So now we have our, like these are sister chromatids. These are identical, made during DNA replication. So this is still in S phase, and then the DNA, volume or amount is double. So then our next step as we go into M phase, into meiosis, would be to, first thing's gonna happen is gonna find their homologous pairs. So now this is where you find their homologous pairs, like this. And then we have crossing over happens. So crossing over happens here on the, you can cross that one over if you guys want. And so what will happen though, is it happens more than once. So when we talked about linked genes, like how close together genes are will determine their likelihood of getting crossed over. So the fact that like this J and this K are so far apart, you can almost guarantee that they're gonna get separated during crossing over. So if crossing over happens more than 50% of the time, you would actually classify like K and J as not linked because they're so far apart. So really like when they're close together is when they're linked. Okay, so now you have, this is called a tetrad because there's 4 sister chromatids. And the next step is where these homologous pairs, are gonna move to the center of the cell. And how they move to the center of the cell, this pair, how it moves here, is independent of this one and independent of that one. It very easily could have been this way, and this one could have been that way. And so this is a second source of variation because every time the chromosome pairs line up, homologous pairs line up along the middle of the cell, it's random. Every single time, and that's going to create variation. So then our next step, in meiosis 1, remember, we're trying to get down to haploid cells, cells with only 3 chromosomes each. So our first step then is to separate homologous pairs. Now why we are separating homologous pairs though, is because we want at least one copy of each chromosome in the gamete. You want one of these with FED, GHI, JKL. You want all of those in the gamete, so you're gonna wanna separate. And here it's like dividing your DNA in half, basically, making sure that each cell is going to end up with one of each type of chromosome. So you separate the homologous pairs. This is where you would have cytokinesis happen. At this point, this is the end of meiosis 1. So you'd have 2 cells, but they still have twice as much DNA, right? So then our next step would be where if these are 2 separate cells, the next part is going to look just like mitosis. So in mitosis, you're going to have these are going to separate, these separate, the sister chromatids, and these separate. So this is going to be one gamete, this will be another gamete, and then here, these are going to separate. Did we do crossing over on those?
Yeah.
Okay.
Yeah.
So these separate, and these separate. So now you have 4 cells that, because of crossing over, are all genetically different. So here you have your 4 gametes, and they have one of each kind of allele, and so they are haploid cells. And then later, during fertilization, you'll take one of these gametes, and it'll join up with another cell, like another sperm or egg, that has the other F, E, D, G, H, I, J, K. So then you go back to diploid. Okay? That's meiosis.